It was in 1952 that Alfred Hershey and Martha Chase performed an experiment that determined the genetic material of a cell. Until this point in time, it was unclear if protein or DNA was the genetic material of a cell. Hershey and Chase worked with the T2 bacteriophage virus because it was simply composed of a protein coat and viral DNA. It was already known that viruses insert their genetic material into bacterial cells to replicate, so they used this knowledge in their experiment. Hershey and Chase performed two variations of their experiment. The first uses radioactive sulfur-35 to grow viruses that incorporate this radioactive element into their protein coats. This allowed them to follow the protein coat through the next steps. The second variation uses radioactive phosphorus-32 to grow viruses that incorporate the phosphorus into the DNA. Both radioactively labeled viruses were then allowed to infect bacteria separately. Then a blender was used to separate the non-genetic factor from the bacterial cells, and the liquid was centrifuged. The heavy, solid pellet at the bottom contained the bacterial cells and the genetic component. The less dense supernatant contained the non-genetic component of the virus. Hershey and Chase found that radioactive sulfur was in the supernatant and radioactive phosphorus was in the pellet. This confirmed that DNA was the genetic material and the protein coat was not. It was, in fact, the DNA that entered the bacterial cells, and the protein coat was left outside the cells. Once DNA was confirmed as the genetic material, the race to uncover its structure began. Rosalind Franklin's experiments using X-ray diffraction provided the experimental evidence to support the structure of DNA that was proposed by Watson and Crick. Photo 51, Franklin's X-ray diffraction photograph of DNA, elucidated key structural features of DNA. Interpretation of the photograph is not straightforward, but Rosalind was the foremost expert in the field at the time. The cross in the center confirmed that the DNA molecule was helical in shape. The angle of the cross indicates the steepness of the angle of the helix. The distance between the dark horizontal bars indicates that a twist of the helix occurs every 3.4 nanometers. And the distance from the center to the top indicates that the distance between nitrogen bases is 0.34 nanometers. It's also important to note that there are some differences between DNA in prokaryotes and eukaryotes. For example, DNA in prokaryotes is naked, the simple strand of DNA that you're probably used to seeing. In eukaryotes, DNA is always associated with histone proteins. The histones are used to tightly pack DNA into the nucleus. Eukaryotes have 50 times more DNA than prokaryotes, so there's a lot to pack in. Eukaryotic DNA is bundled into nucleosomes, which consist of 8 histones that have DNA wrapped around it twice. An H1 histone binds the DNA to the core histones, and the DNA between nucleosomes is called linker DNA. The nucleosomes can coil even tighter to form chromatosomes and solenoids, and eventually coil into chromosomes for division. But overwinding will not allow for transcription, so the coils need to be unwound to allow for transcription. The over- or underwinding of DNA strands is called supercoiling. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter @SciencePet.